we've talked about this idea of a metabolic pathway. One thing leads to another. One thing produces a product that feeds into the next. This is the heart of cell respiration. There are 3 major stages that you need to be familiar with. I highly recommend that you look at videos on cell respiration, animations on cell respiration to help you visualize this process a little bit more. I'm going to give you the nuts and bolts of what's going on, but being able to picture things moving around in your head is much easier, and there are plenty of good animations out there to show you that. So stage 1 is glycolysis. This requires no oxygen for this step, and all we're going to do in glycolysis is break glucose in half. Stage 2 is something known as citric acid cycle, or some of you may know it as the Krebs cycle. I write it as CAC because I get tired of writing out citric acid cycle. But in this process, we're going to break down the rest of glucose as far as we can, and we're going to release CO2 and make a tiny little bit of ATP. Little bit. In this process, the major contributor to this cycle and to this entire cellular respiration process is the citric acid cycle makes very high numbers of NADH and a second carrier known as FADH2. These are electron carriers, remember? And the whole idea with citric acid cycle is it makes a whole lot, or fuels a whole lot of these electron carriers that can then go down to the electron transport chain which is part of this process known as oxidative phosphorylation. Glycolysis, here's the gist. We put in a glucose. We are actually going to have to put in energy. Okay, this is all going to occur in the cytoplasm. So we put in a glucose, we add some energy, 2 ATPs, to kickstart this whole process. We're then going to actually form in this process 2 molecules known as pyruvate. So you've got to follow the bouncing carbons here. So glucose has 6 carbons. We cut it in half. Each molecule has 3. That should be pretty easy for you. We're also going to make some electron carriers. Why? If you break a bond, you release electrons. We need those, so they're going to get picked up by the electron carriers. We're also going to make 4 ATP total. But over here I say that we net 2 ATP. Why do we net 2 ATP? Well, I put in 2, which means I've lost 2 from my system. I make 4. In the end, I end up with 2 that I can use for some other process. And as you might imagine, this ATP production essentially continues to fuel glycolysis. It's unlikely those ATP are going to go anywhere else. They're going to stay right here and continue to fuel glycolysis over and over and over again. So our products, 2 ATP, 2 NADH, and 2 pyruvate, which are 3 carbon molecules. Okay? Cool things about glycolysis, it actually doesn't require oxygen. And since we said it happens in the cytoplasm, there's no mitochondria involved. So this is the earliest evolved form of cell respiration that we know of, and this is what bacteria do. So you do this process, but this is the primary form of energy, or primary source of ATP for our bacteria. Now for you, 2 ATP isn't great. It's not going to power your body. So we're going to have to use another process, that oxidative phosphorylation, to get out more ATP. But for bacteria, this is fine This is enough. This is a terrific advantage to not have to have oxygen, to not have to have mitochondria. The bacteria are small enough and efficient enough that the 5% of the energy that they're getting out of that glucose molecule is fine with them. The citric acid or Kreb cycle, as we mentioned, it's actually going to input something known as acetyl-CoA. Now, the problem is we haven't made acetyl-CoA yet. Okay? So, where did this come from? I told you we ended glycolysis with pyruvate, a 3-carbon chain. So pyruvate is literally 3 carbons. How did we get to, this is a carbon and this is a carbon, how did we get to this 2-carbon CoA thing? This is something that many people refer to as pyruvate grooming. And this actually occurs as the carbon molecules are moving into the mitochondria. So this citric acid cycle is actually all going to occur inside the mitochondria. In order to move that carbon molecule into the mitochondria, we actually have to go through this process of converting pyruvate into acetyl-CoA. Now, this will actually produce a little bit of NADH, right? If you break a bond, release an electron, you're going to make an NADH, an electron carrier. So we'll get a little bit there. Then we finally get into the citric acid cycle. The citric acid cycle, if we're starting with one of these, we actually have 2, right? So we start with glucose, which is 6 carbons. We went to pyruvate, but we went to 2-pyruvate, and now we go to 2-acetyl-CoA, which means we're going to do the citric acid cycle twice per glucose. Does that make sense? You've got to follow the bouncing carbons and the bouncing electrons. Okay, so that's why we have here 2 carbon dioxides are being given off. You're breaking down those last 2 carbons from the glucose on one of these pyruvate derivatives. But we're going to do this twice. So every one of these numbers that you see here is actually times 2 if you want to talk about total glucose contributions. Okay? And you see we make 1 lowly ATP per acetyl-CoA, or we make 2 per glucose. This is not terrific. Again, these are not big numbers. We're going to need more than this. All right, so we've got to look at this generation of NADH and FADH2, the electron carriers, as being really the most important part of this entire cycle. The last step is known as oxidative phosphorylation. So what does it mean to oxidate something? Take away electrons. So you're going to take away electrons that were carried by the electron carriers. You're going to take their electrons now, and you're going to use them in the electron transport chain and power something known as chemiosmosis. So chemio is chemical, okay? Bonds in particular is what we're looking at. Osmosis, we already talked about osmosis. We talked about it with water. I'm looking more now at diffusion. Okay? So let's figure out how this part works. We need to put in oxygen. This process will actually make water in the end. So we have to have plenty of oxygen to do that. We need our electron carriers. We need a mitochondria. Bacteria cannot do this. They don't have the mitochondria. And then we need to set up this gradient. I told you we were going to work on this by facilitated diffusion. We were going to set up an electron or a proton set up this electron gradient with our electron transport chain so that we can pump out hydrogens. Let me show you. This gets complex without pictures, so let me just show you. We need to focus on the oxidative part, which is taking away electrons. We need to focus on the phosphorylation part, which is taking ADP, giving it a phosphate, and making ATP. That adding a phosphate, remember we said, is phosphorylation. So the whole idea is setting up this concentration gradient that is going to make ATP. This is what the mess looks like. So this is inside a mitochondria. Okay, so inside a mitochondria, not just inside a cell right now. So inside, this is the electron transport chain, the proteins that are here. And when we look at the electron transport chain, you see the electron carriers are bringing in electrons and dropping them off. If they're giving up electrons, NADH and FADH2 give up electrons. If they're giving up electrons, that means that they've been oxidized, right? That's our oxidative part. Phosphorylation, this proton gradient that we've set up, we're actually setting up, this is active transport, powered by electrons. So active transport, you know this. Powered in this case, not by ATP, but powered by electrons. And in this process of the electron transport chain, we pump out all these hydrogens because in this process, we're going to actually allow those hydrogens to diffuse back through this transport protein. This transport protein actually has a name. It's ATP synthase. So it's an enzyme. And it literally spins. It's powered like a turbine. And when it spins, when these hydrogens come flying back through, causing this thing to spin around, we actually are going to give enough energy to squeeze ADP and phosphate together and make ATP. Well, we just did phosphorylation then. That's the whole process. So where does the water come in? The water comes in because these electrons are moving from protein to protein to protein, redox reactions the whole way. They get to this final protein and the protein goes, now what? We have to get rid of those electrons so the process can start again and we can keep doing this. Oxygen picks up those electrons. Oxygen is what we refer to in cell respiration as the final electron acceptor. This step right here, folks, is why you breathe in oxygen. Every one of your cells is dependent on it for the efficient conversion of glucose into stored ATP. So in cell respiration, with all of the processes of glycolysis, citric acid cycle, and oxidative phosphorylation, you yield on average 32 to 40 ATP, which makes you anywhere up to 40% efficient in that storage of potential energy. And you make water and carbon dioxide. This is a great review diagram for you. This is where everything moves around and where things are coming in and going out. If you understand this diagram, you would be good with any questions I'm interested in as far as this cell respiration cycle.